जिनिवाते दीनाम् अवित्रवतो
Good afternoon, students. Today I will be talking about enzymes. This lecture has been directed for you to be given a basic knowledge about enzyme, a little bit about the kinetics, and something about the inhibition that we have studied, and a flavor of allosterism. So, why do we want to study enzymes? Now, first obvious reason might be because they exist. That is, for our own curiosity, to get a basic knowledge about what enzymes are. We all know that enzymes have an important role to play in metabolism. Now, in fact, every step of metabolism is catalyzed by reactions, catalyzed by these enzymes, right? And at every step, a particular compound or a substrate is converted to the intermediate, which is again a substrate for the next enzymes. And they work in a consorted manner to generate the entire pathway, right? And this pathway is many times involved in generation of energy for the cells or the organisms, or generation of certain compounds which are used to build up, say, tissues in the body or bones and all those things, right? So it is very important for these enzymes to be regulated, right? So to understand that, again we need to understand enzymes at the first place. Then, as this enzymes converts a substrate to a product, they can sense the presence of substrate, they can sense the presence of product. That is what makes them a very ideal candidate to be studied as a part of analytical biochemistry. Now, once they are used in analytical biochemistry, they can be used as a sensors, what we call them as the biosensors. Of course, in industrial biochemistry, the enzymes can be used in the production of certain compounds of therapeutic importance, right? Or certain compounds which have an industrial importance, right? So again, enzymes become important there. And we all know enzymes are most of the targets of drugs. You take up cancer, you take up any other diseases, the major target that a drug targets is the enzyme. So to understand all these things, we need to know what are enzymes. Before we actually go into the enzymes per se, I'll give you a brief history about how this field has evolved. In 1897, even we all know that Louis Pasteur is known as, he was the one who started the fermentation study, right? So at that point of time, Louis Pasteur said that this fermentation is associated with the yeast cells, and they're associated with the yeast cells which are live, not the dead ones. But later this Edward Buckner, who actually figured out that even the yeast extracts, once you take a yeast, make an extract of it, and if you take that extract and treat it with the sucrose, you can still see the fermentation happening. So Edward Buckner was the first one to say that the fermentation can happen even without live cells, right? So that was one of the turning points where the field started to grow up as to what are those factors which are actually causing fermentation. Alongside, many people like Michaelis and Benton started working upon certain theories which involve enzyme and how do they carry out the reaction. And actually, before this urease was crystallized, everybody, almost many scientists all over the world were in debate as to what is the chemical nature of enzymes. Are they proteins? Are they carbohydrates? Are they lipids? Are they nucleic acids? What are they? So there was a big confusion there, and everybody had their own theories to be given for that. So it was in 1927 when James Sumner from University of Cornell actually crystallized enzyme urease, and once the enzyme was crystallized, its chemical nature could be easily deciphered, and that was a time point when people agreed to the fact that enzymes are protein. And for this, James Sumner was definitely awarded a Nobel Prize because it was a path-breaking discovery in this field. After his wife, Cleland has contributed a great deal in terms of classifying the enzymes into the 6 classes, also devising the kinetic mechanisms to study them, right? So, and the Cleland notation is very famous and he has contributed immensely in this field. Furthermore, we all know that though enzymes function, to be able to understand more in detail about it, we need to know the structure. And the first structure of the enzyme came out in 1968, that was of chymotrypsin. Of course, in 1950s and 60s, John Kendrew could crystallize myoglobin and study the structure. The first enzyme structure came out in 1968, that was of chymotrypsin. Followed by that, it was in 1983, after the advent of recombinant DNA technology, the site-related nucleogenesis came to be available to the scientists, and this became a very important tool to study the involvement of amino acids in the catalysis. So this is a brief history about enzymes. Coming to the most important question, what are enzymes? The first answer that comes to our mind is they are biocatalysts, right? And what do? They catalyze the reaction, increasing their rate. They have a very high reaction specificity, that means they are very specific to the substrate that they convert to the product. They require mild reaction condition. It becomes important because the metabolic intermediates are sensitive to temperature. They may not be viable at a high temperature. Of course, there are organisms which function at a high temperature, right? But overall, biocatalysts, which are enzymes, requires milder reaction conditions. They can be regulated. This comes out to be a very important factor in controlling many things, not only at the whole organism level, but even at the industry level. They are available for regulation. So chemical nature, as I told you, enzymes are mostly proteins. Of course, there are catalytic RNAs which have a catalytic power. So if you bar these two examples of RNA being having a catalytic power, most of the enzymes are protein in nature. And these protein enzymes can be clubbed with a metal ion, or they can have some other organic molecules which can help them to function or to carry out the catalysis. Now if this extra part is a metal ion, we call them as a cofactor. If this extra part is an organic molecule, say for instance vitamins or any other organic molecules, they are called as a coenzyme. Together along the enzyme with the cofactor or enzyme with the coenzyme is together called as holoenzyme, right? And only the protein part of the enzyme is called as a coenzyme, right? So as these enzymes are involved in catalysis of reactions, these enzymes have been classified into majorly 6 subgroups based upon the type of reaction they catalyze. So there are 6 subclasses which involve oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. Oxidoreductase, the name tells you it is involved in transfer of electrons. Transferases, they do a group transfer. Hydrolases, the name tells you again, hydrolysis, a water is involved in breaking of a bond. Lyases, it involves addition of a groups across the double bond, or it involves formation of a double bond by removal of a group. Isomerases involved in isomerization, ligation and condensation of 2 molecules. It is normally an energy consuming process. So based upon this chemical reaction they catalyze, these are the 6 main classes into which you classify the enzymes. Of course, all of these 6 groups, they have a subgroups based upon the substrate they act upon, based upon the cofactor that is needed, based upon the coenzyme if any that is needed, right? We all know that enzymes accelerate the rate of reaction. And how do the enzymes accelerate the rate of reaction? Can anybody tell me that? How do enzymes accelerate the rate of reaction? See, in any chemical reaction, what happens is substrate is converted to a product, right? During the conversion of the substrate to product, it involves an energy barrier. right? And this energy barrier is actually at the peak of this energy barrier is a point where there is a transition state occurs. The transition state is an unstable intermediate of a reaction which has a very high energy, and a substrate has to pass through the transition state to be able to convert it to product. Now, if there is any way, if you can lower this transition state, what are you going to do? You are reducing the barrier. So, if you can reduce this barrier, you can actually make the process easier and faster. That is exactly what enzyme do, or they does. They lower the transition state energy. So this free energy change, delta G double diagram, which represents a free energy change of the substrate and the transition state, is lowered, and that is how enzymes hasten up or fasten up the reaction, right? Alternatively, they also involve orientation and proximity effects. Now, what does orientation and proximity effect mean? If you want to fasten up a reaction or a communication between two molecules, if you get them close together, the chances of collision will increase, right? Secondly, the local concentration can be increased. So that is exactly what enzymes do. They bind the substrate, right? They bind the cofactors needed, and all those things are needed for reaction to happen. They get them concentrated in a small space. And what is happening? They are in the close proximity, and that is what enhances the rate of reactions. Or again, the other part is orientation. Now the reaction has to happen, and if a particular bond is to be attacked, if it is oriented optimally, the barrier, the time that it would take for the bond to break will be slower, and ultimately the reaction will get faster. So all these three together helps in fastening up the rate of reaction. But the major contribution comes from transition state binding or lowering the transition state energy. Is that okay? Now, why energy is required to reach a transition state lower? Let us see this animation. It tells you a little bit about it. It stabilizes the transition state at the active site. Alright. It expels the water molecule, which might form the bondings in the transition state. It places the reactive group optimally so the reaction can happen. And at the end, there is a coenzyme which not only is involved in binding of the transition state but also actively takes part in the reaction. So all these four factors together helps to fasten the rate of reaction. Is that okay? Now to further reiterate this point, just consider this example. This example has been taken from Lengen, Nelson and Cox, which talks about a very hypothetical enzyme called stickase. Okay. Now what is a stickase? There is a substrate which is metal stick. It has to be broken down to two sticks. That's a product. The substrate has to be converted to a product, which involves a transition state of bending. Alright. So there is a transition state which is a bent state which is unstable. Now imagine a molecule that just binds a substrate, which is complementary to the substrate. Right? Say for instance it's a metal stick, so consider a magnet. Magnet will bind to that stick, but will it break it? No. Now consider this alternative molecule, wherein the structure resembles the transition state. So once the stick binds to this, it forces the stick to form a transition state and ultimately bends it. So what is it doing? It's lowering the transition state energy, bringing it down and fastening up the reaction. Is that right? Let us have a look at a comparison as to what is the order of magnitude to which the enzyme increases the rate of reaction. Now all these enzymes, carbonic anhydrase and all these enzymes catalyzes a specific reaction. In this particular table you will see a non-enzymatic rate constant and an enzymatic rate constant. So in the absence of any enzyme, carbonic anhydrase converts bicarbonate to CO2 and water, right? Or carbonic acid to CO2 and water, and vice versa. It even catalyzes a reverse reaction. It can. So, in absence of any enzyme, the breakdown rate is 10 to the power minus 1. Whereas once the enzyme is present, the rate enhances by 10 to the power 7 folds. So, you can see the order of magnitude to which the enzymes increases the rate is considerably high, right? Now, what are the factors that affect the enzyme? Substrate concentration, of course, because a substrate is involved, the enzyme is involved. So, concentration of any of these two will affect the rate of reaction. Third would be because these enzymes are protein in nature, right? So they might be sensitive to temperature, correct? And also the another component that is involved in stabilizing the structure of protein is pH. So all these four factors can affect the rate of reaction or the enzyme catalyzed reaction. Let us see how the substrate concentration will affect. You keep increasing the substrate, the rate increases. After a while, the rate saturates. Why does it saturates? It's because when you initially increase the substrate, because of a higher concentration, the position is frequent, right? But after a while, the reaction rate levels off. Now that happens because all the active sites of the enzymes are not saturated and are involved in catalysis. So no more enzyme is free to carry the condition anymore. Is that clear? Now coming on to the next part, that is enzyme concentration. So consider this first graph, which is here written as 1x. Initially this is the when you increase the enzyme concentration or keep a concentration of enzyme in 1x, you see a falling plot with the increase in substrate concentration. So after a while the reaction rate saturates. But when you increase the enzyme amount, the amount of active sites have increased, right? So they will take more substrate to saturate, correct? So the saturation that it takes, the time it takes to saturate will be longer, or the concentration of substrate required to saturate will be higher, right? Now in this case, what is happening? The maximum velocity that is reached changes. Okay, the maximum velocity that is reached changes with the function of enzyme concentration. So that is how the enzyme concentration affects the reaction. Correct? Now look at the temperature. If you increase the temperature, initially the rate will increase. It will reach the optimal reaction rate, after which the rate will drop. Alright. And why does this happen? It happens because once you increase the rate, temperature, the enzyme denatures. The enzyme loses its structure, which is actually responsible for the activity. Is that okay? Fine? This points are clear for you all? Let us see how pH affects the rate of reaction. If you change the pH, what are you actually doing? You're kind of changing the H plus ion concentration in the solution, right? Now, because it's changing the H plus ion concentration, it may disrupt the bonding, because of which the 3D structure might get changed, and that will affect the rate of reaction again. So, what is the optimal pH? For most of the enzymes, I would say the optimal pH comes to around 6 to 8. Right, that is the physiological pH of the cell, what we see. Of course, there are extremes, enzymes which work at a very alkaline condition and which works at a very acidic condition. And both these enzymes are of industrial importance again. Okay. So, we know that substrate concentration affects the rate of the reaction, and the normal substrate saturation plot will appear something like this. Correct? So after you have increased the substrate concentration to a high value, the reaction rate levels off. And this plot is actually known as a Michaelis-Menten plot, right? So Michaelis-Menten plot is a function of change of reaction velocity with respect to substrate concentration. Please take a note, it is referring to initial velocity and not velocity alone. Now what is the difference between a velocity and an initial velocity? Consider the rate of reaction, if you are monitoring a rate of reaction as a function of time. Alright. Right? The moment you start the reaction, that means you take a cuvette, you add all the components, buffer, substrate, and then in the end you add enzyme. Right? So the moment you add all these components and mix, that is the time when you start the reaction. And if you start monitoring the rate of the reaction from that point of time, what you see is initially the rate is very high and then it plateaus off. Why does this happen?
Because the substrate binding active sites would not be present. Once the reaction starts, the active site would be maximum, and once the enzyme binds to the substrate, the active site decreases.
Let us consider this reaction. Enzyme plus substrate giving enzyme plus product. Initially when you started the reaction, say for instance you had 100 molecules of substrate with you. Once the time passes, as the time passes, the concentration of substrate will drop 90, 80, 70, it will keep dropping. If we reconnect the factor that affects the enzyme reaction, the concentration of the substrate will affect. Correct? So that's the reason. Initially the substrate concentration is very high. So you have a very high rate. And then it keeps dropping off because substrate is getting limiting. And at this point of time substrate is over. Now if you measure the rate here, you will see there is no rate happening. There is no reaction happening. If you measure the rate here, it's not correct. You will have to measure the rate somewhere here. Now if this time is in minutes, you have to actually scale down to seconds. So you have to read the rates in the second time scale. You have to zoom in the time. And those kind of kinetics are actually called as fast reaction kinetics. Now let us zoom in here and see what happens. Now we have got to seconds. Alright, say for instance this was 0 to 2 minutes, 120 seconds, 60, 30. The rate is initially maximum for say 60 seconds and then it starts dropping. So this is the time point where you have to actually measure the rate. And this rate is known as the initial velocity, where the rate of reaction is not dependent upon any other factors, wherein the substrate concentration change has happened to the least possible. So whatever substrate concentration is added, you are measuring the rate at exactly that concentration of substrate and not at any other. Fine. So for a convention, the substrate to product conversion should be less than 10%. Okay, substrate to product conversion should be less than 10%. And when you monitor the rate under this condition, that is what we term it as the initial velocity. So those two terms are different. Is that very clear? So once you plot initial velocity as a function of substrate concentration, you see that initially it rises, then it plateaus off. Please mark there are certain characteristics of this Michaelis-Menten plot. The initial phase that you see, you increase the substrate, the rate increases. You increase the substrate, the rate increases. This kind of reaction kinetic is a first order rate. Correct? It's a first order reaction. Look at this part of the graph. In this part of the graph, the rate of the reaction is almost not dependent upon the substrate concentration. Correct? So it's a zero order rate. Correct? It's a zero order reaction. Then how much ever you increase the substrate, there is no change in the rate, or hardly any change. Between this first order and the zero order, this part that you see is known as a mixed order phase of the reaction. So there are three phases in a microinverting curve. First order, mixed order, zero order. And this particular part is also, this particular part becomes almost insensitive to substrate change. However, you see the Vmax, what we call it as a maximum rate achieved by a reaction, is not somewhere here. You know why? If you technically see, the Vmax should have been here, correct? Because this is the maximum rate that you see. But the Vmax is not higher than this. Why? Because this particular graph will reach this space at infinite substrate concentration. It will tend to it, but will never reach it. Right? We always say this x tends to 0, but x is not equal to 0. It is close to 0 but not 0. Limits, right? Same way, the reaction rate is tending to maximum velocity but not reached so far. That is one drawback of Michaelis-Menten graph to be used to determine the kinetic constants. Understanding this? However, you can still determine, because once you don't know the Vmax properly, your Km value also may not be very correct, accurate. Understanding this? I am not getting to the derivation part of it, but I am sure everybody is familiar with this Michaelis-Menten equation which states velocity equals Vmax into substrate concentration divided by Km plus substrate concentration. Is that fine? So, another plot that is there is Landweber-Berg plot. It's a double reciprocal plot. Why is it double reciprocal plot? Because both the x and the y axis has a reciprocal of the variables, right? So because of this, it's 1 by Vmax and 1 by S. And in this particular plot, the advantage is the intercepts directly give the value of Km and Vmax. However, this plot also has some disadvantage, which I'll talk to you about it a little later. Let us look at the catalysis. As the enzyme catalyzes the reaction, there is a mechanism that it follows. Correct? So there are four mechanisms on the basis of which a reaction is catalyzed. One is acid-base catalysis, covalent catalysis, metal-ionic catalysis, and electrostatic interactions. Okay. We look for the few of those. In the acid-base catalysis, what happens? Because enzyme has an active site, when there are amino acid groups present, these amino acid groups can serve as a donor of protons, or it can serve as a acceptor of protons. And this particular fact can be used to catalyze a reaction. So where do you see the acid-base catalysis happening? One example is hydrolysis of peptide. In this case, again, tautomerizations, the conversion of one form of glucose to the another form of the glucose. That also involves hydrolysis, this acid-base base conversion. And aspartic acid and all these amino acids are known to be involved in the acid-base catalysis. This is what exactly I was referring to, meta-rotation of glucose from alpha form to the beta form. Coming to the next part, that is the covalent catalysis. In the covalent catalysis, the name tells you there is the formation of a covalent bond. Alright, and this covalent bond formation will be at the transition state. So there will be a transition state in which the substrate will be covalently bound to the enzyme. Alright, and mostly in this case, hydroxyl groups containing amino acids, sulfidyl group containing amino acids, imidazole containing group amino acids are involved in the covalent catalysis. And this covalent catalysis can be a electrophilic or a nucleophilic. I'm sure everybody understands the term electrophilic and nucleophilic, right? Coming to the next part is metal ion catalyst. In the metal ion catalyst, the metal ions will be involved in the catalysis. Now please take a note, there are two terms involved here. Metalloenzymes and metal activated enzymes. Metalloenzymes and metal activated enzymes both require metal ion, but they differ in their the metal part. How do they differ? In metalloenzymes, the metal component is tightly bound to the enzyme. It cannot be dissociated easily. Whereas in metal activated enzymes, they are loosely bound. So they are dissociable. Right? That's why the difference between the two. And in this case, how do the metals help in the catalysis? They help to bind the substrate and orient them for catalysis. Remember, the rate enhancement happens because of orientation also. At this point of time, metal ions help the substrate to be oriented correctly. Right? Alternatively, there is a redox reactions also happening. And it also stabilizes the transition state. Because metal ions will mostly carry a positive charge, so if any transition state involves a negative charge state, that will be stabilized by the metal ion. And that is how the catalysis happens. Right? This is an example in which the transition state here is stabilized by manganese. Alright. So because it is stabilized, the energy of transition state component lowers down and the rate increases. Is that okay? Similarly, carbon kinetics, in this the zinc is involved in the reaction and it stabilizes the charge residues over there and it catalyzes the reaction. Similarly, ADP, then magnesium is involved. In fact, all this information is actually helpful for us into the research and experiment. For instance, if you are doing certain PCRs, or certain restriction digestion, in that cases, if you want to stop the reaction, because they act on DNA and RNA, they actually need these divalent ions, right? Because it will be shaping the charges. So you add a ADT. What will it do? It will chill it up the metal ion. The moment the metal ion is chilled out, the reaction is stopped because the reaction is dependent on the metal ion. So all this information helps you to even design an experiment or stop a particular reaction or start the reaction. Right? Now we have seen how the enzyme catalyzes the reaction. We have seen the plots. We know that this reaction is a dynamic process. It keeps happening, right? So because it's a dynamic process, it will involve the rate. That also we have seen. And study of all these things are known as kinetics or the enzyme kinetics. Now let me emphasize one point here. We see that enzyme is increasing the rate. So what is it doing? It is increasing the rate. Okay. But does it affect this? You know what is this called as? Equilibrium constant. What is equilibrium constant? It's a constant of it's a ratio of concentration of product and the substrate or the reactants. Correct? But does it affect the equilibrium constant? Enzymes affect the rate, but they do not affect the equilibrium constant. In fact, nothing will affect the equilibrium constant unless until you make some external intervention of temperature or change certain factors in the reaction. Otherwise, enzymes can't alter the equilibrium constant. This is very important to be noted. Coming back to the candidate parameters, I talked about line to a work plot. There are many more plots available: ADRC plot, Hans plot, direct linear plot, and even certain statistical measures or computational ways you can actually determine the kinetics. This is a line to a work plot. Now, looking at this example, what is the drawback to this plot? The drawback to this plot is you are taking the reciprocal of substrate and you are taking the reciprocal of rate. Correct? Now, just imagine this concentration of substrate is we choose. To get this particular graph, we have to do an experiment. Right? We all know. We have to do an experiment. And what is an experiment? It is an experiment to determine the rate as a function of substrate. Am I right? So you keep changing the substrate to determine the rate. Is that right? And that is how you get a plot of Michael's method. Now consider this example. You take the substrate as 1, 2, 3, 4, 5 and 10, 15, 20. Now what you are going to do, you are taking a reciprocal of it. When you take a reciprocal of this, the points at a higher 1 by s. Higher 1 by s value will mean what? The substrate is very low. And understand that once you are lower in the substrate concentration, when you are going lower in the range of concentration, the chances of error increases. The chances of error increases. So because of it, what happens? You have a very high error at the lower substrate concentrations. Whereas in the higher substrate concentration, the errors are very small because accuracy and precision is better. Right? The volume of solution that you are pipetting are much more in control in this range. And what happens because of this? Your plot can run like this, your plot can run like this, your plot can run like this. Alright? Because of this, you can wrongly estimate a Kn and Vmax values. Now how do you correct it? What you can do is you can do a weighted average. I will go to the details of this. Weighted average is a method wherein you give the same weights to all the plot points in the graph and then do a plotting. So that is how you correct it, but the disadvantage is this. Understand? So again in line to work, you cannot accurately estimate the kinetic parameters. But yes, line to work is very essential in understanding one thing, the nature of inhibition. We will come to that later in our presentation. Now ADRC plot. Now ADRC plot is a plot of velocity versus velocity by substrate. This is again a linear plot, but it has the same problem. The problem is in both the axes we have velocity being used, and velocity is a dependent variable. So it is not a very good idea to have a dependent variable on both the axes, because we are estimating a dependent variable, right? So if you are estimating that wrongly, we are affecting it, we are increasing the error by twice. Not only your y-axis is affected, your x-axis is also affected. And again this plot becomes a little disadvantageous to estimate the kinetic parameters. Hans plot is considered to be one of the better ones in estimating the reaction parameters. It's because it's a plot of substrate to velocity ratio versus substrate. In this case, you have a dependent variable in only one of the axes, whereas the other axis is an independent variable. You are actually adding the substrate, right, in the machine learning. That's why Hans plot becomes a little better to estimate the kinetic parameters. Of course, nowadays, hardly few people use actual manual plotting. We always go for statistical analysis and plotting and getting the data out using the compilation softwares. There is one more plot called a direct linear plot. Now, what is a direct linear plot? Direct linear plot is you have an axis of substrate and you have an axis for velocity. For every substrate concentration, you will get a value of velocity. So, for instance, if you take 40 units of substrate, 4 units of substrate, and if you get an activity of say 5, you place a dot on the x-axis at 4, you place a dot on the y-axis at 5, and you connect the lengths. Now do the next estimation. Substrate at 10, what is the rate? That's the rate. You connect the dot and keep connecting. They will intersect at one point, and that intersection will give the Dmax and the K. But what is the problem here? If the intersection is precise single point intersection, you can estimate, but there are chances that you might see the intersection something like this. In this, the range will be this much. You are actually increasing the error here. The intersection is not a pinpoint intersection. Right? So that's the limitation of again direct linear plot. So all these plots are used in the determine the kinetic parameters. Coming to the other part, we have talked about the enzymes which are following Michaelis-Menten kinetics. So, this plot is a Michaelis-Menten plot, right? Right? This is a Michaelis-Menten plot of rate versus substrate concentration. Now, not necessarily all the enzymes will behave like this. Some enzymes might behave like this. You change the substrate, they don't change in the rate. You change the substrate, they don't change the rate. You change the substrate, they don't change the rate. A small change in the substrate will drastically change the rate. This plot is known as a sigmoidal plot. And this sigmoidal plot occurs for the enzymes which are allosterically regulated. Now, what does allosterically regulated means? They follow. So, if you take it as an enzyme, normally enzymes have an active site. Allosteric enzymes will have a site other than the active site. Right? So, if you get a distance, say somewhere here. So, binding of one molecule here will have an effect here. Those are the allosteric enzymes, and they are very important because these are the allosteric enzymes which are involved in metabolic flux, phosphofructokinase. They are under the control of many intermediates and many cofactors and coenzymes, which senses the energy level of cells. These enzymes are also important for on and off. See, for this range of substrate, the rate is completely off. For this range of substrate, the rate is completely on. So they are very essential to set the metabolic switches. Understand? So these are the allosteric enzymes, and they can be regulated by various ligands. If these ligands activate them, alright, so they are called a positive activator. What will happen if they activate say for instance, if you consider an allosteric enzyme which follows a sigmoidal plot, alright, and if you add a component that is enhancing the rate or increasing the affinity of enzyme for the substrate, the plot will shift to Michaelis-Menten. They are called as positive effectors. Alright, and if there are certain molecules which are inhibiting the rate, then the plot will become further inward. They are known as negative effectors. So positive effectors will increase the affinity of the enzyme for the substrate and increase the rate. The plot will become Michaelis-Menten. The negative effectors will bind to it, reduce the affinity and reduce the rate. the plot will become further sigmoid. And that's when the enzyme rates are regulated. Let us see that because these reactions are there, they can be inhibited as well, right? So this inhibition is can be of various types. One of these types is competitive inhibition. Competitive inhibition, there is a competition involved, right? There is an enzyme which binds to substrate on the ES complex and that forms a product, and the enzyme is regenerated in the reaction. Now, if a molecule is added that also binds to the enzyme, it will compete with the substrate for binding. And what is going to happen? If the inhibitor binds, then no reaction takes place. So, what is going to happen? It is reducing the affinity of the enzyme for the substrate. Correct? And consequently, what is happening? The rate of reaction is also dropping. Am I right? So if you look at the plot, the moment you keep increasing the inhibitor, the affinity for the substrate decreases, and the amount of substrate that is needed to reach the saturation will also be higher. However, the Vmax will remain unchanged. Why will Vmax remain unchanged? Because once you increase the substrate concentration to a higher amount, it will finally replace the inhibitor back and the reaction will take place. Correct? Understand this? So that's what competitive inhibition is. So this plot, if you see, it's a reciprocal plot. That's a Line-Weaver plot. And that is where the importance of Line-Weaver plot comes. To understand the nature of inhibition directly by looking at the plot. The moment you look at the plot, you see that Vmax unchanged, you see that Am changed, you can say it's a competitive inhibition. Correct? Next one is the uncompetitive inhibition. In the uncompetitive inhibition, what happens? The inhibitor binds to ES complex and not to enzyme alone. Correct? So because of this, what happens? Whatever ES is formed, it is ultimately from ES side complex and no reaction will occur. Correct? If you look at the plot, what is happening? You are increasing the inhibitor, the Vmax is increased. I'm sorry, the Vmax is decreased and the reciprocal is increasing. Correct? Why the Vmax is increased? Because whatever ES complex was formed, inhibitor is bound to it and no reaction has happened. Correct? The maximum velocity is never attained. So the moment you add more inhibitor, more ESI will come, the maximum velocity will drop further. Correct? But see one interesting fact. The reciprocal of Km is increasing. The reciprocal of Km is increasing. Correct? But it's on the negative axis. So what is happening? The Km is actually decreasing. Km is decreasing means what? The affinity of the enzyme for the substrate has increased. How can that happen? A inhibitor has actually increased the affinity of the enzyme for a substrate but dropped the rate. Can anybody explain why? Let us see this. When the enzyme binds to substrate, it forms ES complex and this is in equilibrium. But when the inhibitor binds, it forms ESI. So ES concentration is dropped. The equilibrium will shift to right. More enzyme binds to substrate. Again ES is formed, again it forms ESI. Again the concentration of ES is dropped down. More enzyme binds to substrate. That is the reason the Km has dropped, affinity has increased when the rate has dropped down. Again, this is a very interesting feature of this uncompetitive inhibition that one needs to understand. The last one of the most possible inhibition that you see is non-competitive inhibition. We have seen a competitive one when the enzyme binds only to E. We have seen a uncompetitive one when the enzyme binds to ES. The last one is non-competitive. Whether substrate is present or not. I will go and bind to the enzyme. That's the non-competitive. It's not in the competition at all. It is going to bind to the enzyme, irrespective of what in the state it is. So what happens to the plot? In the plot, the Vmax will drop. Vmax will drop, correct? Because ES has found ESI, but Km has unchanged. Why? Because EI does not interfere with the binding of enzyme and substrate. It does not interfere there at all. It is not interfering in this part. It is interfering in this part. Correct? So because it is interfering in this part, the Vmax is dropping. But the Km has unchanged. So let us reiterate the entire point. In uncompetitive, we have a change in Km but not in the Vmax. In uncompetitive, no changes. Right? It is actually affecting both. If you go back, it is actually affecting both. It's affecting this part, it is affecting this part. So the Km has to change, Vmax has to change. Right? And in non-competitive, it does not interfere with this part, it interferes with this part. So you have a change in Vmax but not in the Km. Is that very clear? So this slide will sum it up all. It binds to the P enzyme in competitive. Non-competitive, it binds to the enzyme irrespective of substrate binding. In uncompetitive, binds to ES complex, and that is how this is the major inhibition classes that you see. But let me tell you, practically, most of the enzymes you will find them following a mixed inhibition. There are plenty of cases of competitive inhibition, what we call as true competitive inhibition. But at the practical level, you will observe that majority of the inhibitions are mixed inhibitions. We already discussed about allosteric enzymes and modulator. This slide is just to tell you that there is a catalytic unit, which is here, and there is a catalytic subunit, which is actually the allosteric site, where the regulatory elements will bind. So the regulatory element binds, and it increases the affinity for the enzyme, for the substrate, we call it as a positive modulator. If this element reduces the affinity, we call it as a negative modulator. Right? And everybody recollects, if it's a positive modulator, the plot will shift to Michaelian content. If it is a negative modulator, the plot will become further sigmoid. You see this? It's shifting more towards Michaelian content. And this modulation can not only happen because of the allosteric site, but it can also happen when there are multiple enzymes. So binding of a substrate to one subunit increases the binding for the second subunit. That happens in which protein? If there is an example of a protein which has 4 subunits, and once the component binds to one subunit, it increases the affinity for the others. Hemoglobin, right? That also follows the sigmoidal saturation. So that's all for this lecture. Any questions? Anything I would like to clarify? So what have we studied in this particular section? We have studied water enzymes. You have a question?
Yeah, so when does the mutation takes place? In case of now mutation of glucose is there. So under what condition does the mutation take place?
It takes place normally in the acidic conditions. Alright. So once the mutation is also possible in solution, but you have to make the conditions a little extreme because it's an acid-base catalyzed conversion. So in case of enzymes, if you see the pH of the solution does not matter. What will matter is the pH of the microenvironment. And let me tell you, we all know about this amino acids having a pKa value. Correct? But the pKa value of amino acids in isolation or in a protein will be very different or might be different from the pKa value that you will exhibit at the active site. A histidine which is known to have a pK of around 6.4 can also have a pK of 4 at the active site. Now, why does that happen? Because there might be hydrophobic groups present which might prevent the charge formation, that will prevent the dissociation of the proton, and the histidine might remain in the NH form and not get protonated. Any other questions? That's all. Thank you for your attention.